Hello, IB Bio students. All right, so today we are going to start higher level topic 10.2, and that will be inheritance. So notice that inheritance is covered by different core content here. Today we're going to be looking at dihybrid crosses. Then we're going to look at linked genes and recombinants together. And then we're going to learn how to do a chi-square test analysis, which you started in SL, but we'll continue in HL. And then we'll look at polygenic traits. So today we're going to do dihybrid crosses. And the nice thing about dihybrid crosses is that it's pretty much nothing new from what you learned back in the 9th grade and you reviewed in SL material. All right, so here's the essential understanding. Unlinked genes segregate independently as a result of meiosis. So we really need to understand what an unlinked gene is. Because when we start learning about linked genes, we're going to really need to understand the difference, okay? Because linked genes is something you haven't done before. Or maybe you have, but only sex-linked. All right, so according to the law of independent assortments, remember that Mendel formulated different laws, the law of segregation, the law of independent assortment. He said that pairs of alleles are inherited independently of one another if their gene loci are on separate chromosomes. So if the allele or the gene loci of that allele is on a different chromosome, then these genes are said to be unlinked. This is due to random orientation, which we saw back in the last topic 10.1. So this is due to the random orientation of the homologous pairs during metaphase 1 of meiosis. The independent segregation of these unlinked genes results in a greater number of potential gamete combinations, as well as a greater variety of possible phenotypes. So back in the 9th grade, you looked at the difference in these complex inheritance patterns, but really all you focus was, the focus for 9th grade and 11th grade was Mendelian inheritance patterns. So you looked at Mendel's experiment and what he did when he crossed pea plants and he crossed monohybrid versus dihybrid. So what is the difference here? Let's look at the picture. So here you have inheritance of a single gene versus 2 genes, but they are both unlinked. That means that they are on a separate chromosome. And again, we really have to visualize this. Okay. So here we just see one gene, and we call that gene A, and perhaps that gene is could be a pea plant characteristic for color, or it could be for brown eyes, blue eyes, whatever you want to call it, right? So one gene for, let's say, hair color has 2 possible alleles. And these 2 possible alleles, one can be dominant, the other one can be recessive, or they can both be dominant, or they can both be recessive. And these allele combinations are found in a different chromosome. So that's what we mean by them being unlinked. They're on separate chromosomes. Now, they both code for one gene, though, right, which is hair color. So anytime we have a dominant allele, we note that with a capital uppercase letter. And when we have a recessive allele, we note that with a lowercase letter. Okay, so then that means that when these chromosomes undergo independent assortment and they form different gametes, these gametes are going to end up with potentially 2 allele combinations, right? You can end up with this combination or this combination. Okay, so in other words, what this is saying is, let me erase this. What this is saying is, what happens when you cross this with this, right? And you know how to do this, right? You know how to do this because you just do a monohybrid punnett square. Okay, we have one gene and two alleles on two separate chromosomes. And then you know that the potential offspring, or the F1 generation, could potentially look like this, right? And all these are probabilities. So you're going to end up with a 50% chance of a heterozygous offspring, or 25% chance of a homozygous dominant or homozygous recessive offspring. And that's because of the law of independent assortment, right? These two alleles separate independent of each other, so they don't go together. All right? So when we think of one gene, we think of a monohybrid cross. Okay? Now, in contrast, you can have two genes which are also unlinked, so they are on separate chromosomes. And in this particular case, you have two genes. So notice that you have a big B, little b, and you have a big E, little e. But again, you have different allele variations, right, for that particular gene, but they happen to all be on a separate chromosome. So we call these unlinked, okay? So in this particular example, we have perhaps a a combination of this, right? And then you cross that with something else, okay? So these gametes here, right? These gametes here are going to recombine independently of each other. So we're going to go over the FOIL method in just a moment, but you remember doing this, right? Because you know that this could give you a possibility like this, big B, big E. It could give you a possibility like this, big B, little e. Where is it? It could give you a possibility like this, little b, big E. Or it could give you a possibility like this, little b, little e, right? So you have four possible allele combinations in the gametes. So when you cross this potential parent, you end up with four possible allele combinations. And then when you cross it with another parent, right, that other parent might have, if that other parent is also heterozygous, that other parent might also have four possible gamete combinations. Okay, and this is what's going to give us a dihybrid cross. Right, dihybrid means two, so you have two genes that are unlinked on separate chromosomes, which could potentially give you four different combinations. Okay. Now, the punnett square for a dihybrid cross is going to look completely different than a monohybrid cross, right? These are the bigger ones. So, how do you complete an analysis of a punnett square? And that's your application. So you need to be able to do dihybrid punnett squares. And I have a packet for you where you're going to practice all these. Now, the easiest way to work out a dihybrid punnett square is to first determine what the possible gametes are going to be from a particular parental genotype. And I like to use the FOIL method. Now, here in BioNinja, they give you another possibility, which is called the claw method, which is like you have to draw a claw like this, and then you do the same thing. First, outer, inner, last, right? But I don't know, whatever works for you. I like the FOIL method. So just what I did in the previous example, right? These are the two genes, right, with four possible alleles. So how are they going to recombine via the law of independent assortment? So they're going to recombine independently of one another. So the first alleles could give you one combination. The outer alleles can give you one combination. The inner alleles can give you one combination. And the last alleles can also give you one combination. Right? So you have four potential gamete combinations, and this is the FOIL method. So here are step-by-step instructions. Oops, what happened here? Oh no, sorry about that. lost my place there. Here we are. All right, so here we have step-by-step instructions on how to complete a dihybrid cross, right? So first you designate characters to represent the alleles. Capital letter for a dominant allele, lowercase letter for recessive allele. You write down the genotype, and remember the genotype is the allele combination, and the phenotype for the parents. Remember the phenotype is the physical representation of the parents. Always pair the alleles from the same gene together. So A goes with A and B goes with B. Not like this, right? Write down all the potential gamete combinations using the FOIL method, and then use a punnett square to work out the potential genotypes. And for a dihybrid punnett square, the punnett square is going to be pretty big, and I'll show you in just a moment. And then when you're done, you're going to write out the phenotype ratios of the potential offspring. So here is what an example of a dihybrid cross looks like. Notice that a dihybrid cross is a 4x4 punnett square. Okay? So let's say you have a yellow round seed and you cross that with a green wrinkled seed. So this is what Mendel saw in his pea plant experiments. Okay? So these are both homozygous genotypes. So then that means that you're only going to get one possible gamete here. Same thing here. You're going to get only one possible gamete here. So you know that the F1 generation, or the first filial generation, is going to be all yellow round, right? Considering that yellow, big Y, is dominant over over green, and round is dominant over wrinkled, little r, okay? But what if you do a cross between these two F1 offsprings? So that's what we call self-fertilization. So you're going to cross this with this, right? And you've seen these before, right? So first you have to do your FOIL method. First, outer, inner, and last, right? And that's going to give you all your 4 different gamete possibilities. Big Y, big R, big Y, little y, big R, and little y, little r. So this is what we call, right here is what we call independent assortment, right? The alleles are independently assorting for that particular offspring. The allele is also independently assorting for that other parent. So here we're crossing 2 parents from the F1 generation, and then this is what you get. Okay, so these are all the F2 potential offspring, so filial 2. So then you have to count, right? And you have to count. First you have to, if you want to figure out the genotype, then you have to look at all the possibilities. Now, I'm already going to tell you that anytime you do a cross between 2 heterozygous, which is what you have here, you're always going to get a 9:3 to 3:1 genotypic ratio. Okay, 9:3 to 3:1. So might as well memorize that. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9. Sorry, not genotypic ratio. Sorry, phenotypic ratio. I'm so sorry about that. Okay, so that means that you're going to end up with 9 yellow rounds, and then you're going to end up with 3 yellow wrinkles, and then you're going to end up with 3 green round, and then you're going to end up with 1 green wrinkled. Now you could potentially list out all the genotypes, but we usually don't ask you to do that in a big dihybrid cross like this one. Okay, so the phenotypic ratio of 2 heterozygous dihybrids is always going to be 9:3 to 3:1. All right, so the skill is to be able to work out the punnett squares. So we're going to work out the punnett squares now. So below are two examples involving dihybrid crosses. So the first thing I suggest you do is read, right? What is this asking for? And then I always like to outline what I'm reading. So let's see. In sheep, the allele for black wool is dominant. So black wool is capital B over the allele for white wool, which is recessive. Similarly, the alleles for horns is dominant over the allele for being hornless, little h. So if it tells you that pure breeding horned sheep with black wool are crossed with pure breeding hornless sheep with white wool, pure breeding means that they're homozygous. So then this means that a pure breeding horned sheep with black wool, right, is going to be big B, and then it's horned, so big H, big H. Now, it doesn't matter if you wrote it like this because you're going to end up getting the same thing anyways. Doesn't matter how you write it, okay? And then you're crossing that with a pure breeding hornless sheep that has white wool. So that one's going to look like this. So this is going to be a pretty simple dihybrid cross for question A, right? Because it says state the genotype and the phenotype for the F1 individuals. So we know that the possible gamete here for this parent is big B, big H, and the only possible gamete for this parent is little b, little h. So let me expand so you can look at the answers here. And this is what we have so far, right? We're crossing this black, what's it called? Black wool horned sheep with this white wool hornless sheep, right? And because the parents can only have one possible gamete combination, that means that you don't even have to do a dihybrid cross, right? You know that you're going to end up with 100% black horned. But now it tells you that you're going to take two F1 offspring and mate them together. So then that means you're going to take two of these F1 generations, B, and you're going to mate them together. Now here is like the example that we just saw. Right here, you're going to end up with 4 possible gamete combinations for this parent, 4 possible gamete combinations with this for this parent. So as we continue down, you have to do your 4x4 punnett square. Okay, and notice that they just want you to calculate the phenotype, right? So the phenotype again is the physical expression. So it's just like, do you have black hair? Do you have blonde hair? Do you have blue eyes, right? So anytime you cross two heterozygous in a dihybrid cross, you're going to end up with a 9:3 to 3:1 ratio, okay? So if on a test you get a question like this, right, and it's maybe a multiple choice question, you don't even have to work out the punnett square, because you're always going to get 9:3 to 3:1. So you can save some time there. All right, now let's look at the other problem. In this particular problem, you have cats, and you have the allele for gray fur is dominant over the allele for beige fur, and the solid coat is dominant over the allele for a striped coat. So you are doing a cross between a pure breeding solid beige cat. So what is that going to look like? A pure breeding means it's homozygous, beige cat, and solid. So that's what this cat's going to look like. And it's crossed with a pure breeding striped gray cat. So if it's gray, it's going to look like this. And if it's striped, it's going to look like little s. And I hate when they use s because it's hard to tell the difference between a capital S and a lowercase s. But try your best. All right, so just like in the first problem, right, these parents can only end up with one possible gamete combination. So then that means you don't even have to do a dihybrid punnett square. You can just cross this with this, and you're going to end up with your F1 individual, right, which they are all going to look like this. So I don't even have to do the cross. I know that all my F1 individuals are going to look like this. But then I have to calculate the phenotypes resulting from a cross between a pure breeding solid beige cat. So the cat is beige and it's pure and it's solid, which is big S, and my F1 offspring. So you really have to pay attention to what the question is asking. So you're going to be crossing now this guy with one of the F1 generations. So back here, you crossed two F1 generations, but not here. All right, so just pay attention to what the problem is asking. So when you expand here and you look at your answers, right? This is kind of what I had, right? So you know that the F1 generation is going to be big G, little g, big S, little s. And when you cross that with the other pure breeding solid beige cat, this parent can only give you one potential gamete, which is this one. I mean, so you can do 4 possible gametes here, but it's just going to be a waste of time, right? So you're crossing this parent gamete with these 4 possible combinations from the F1 generation. And that means you're going to end up, and notice that here they're asking for phenotype, right? So they're asking which ones will be solid gray, which ones will be solid beige. Would there be any, what's the other, striped gray or striped beige? But here you see that you have 2 solid grays and 2 solid grays. So 50-50 phenotypic ratio. So we're going to practice these, and I do have a genetic packet, review packet for you guys. So after you're done with your genetic problems, I will also post the key for you to check your answers and see how well you remember these dihybrid crosses. So nothing that we've done here as far as dihybrid crosses is new, right? Everything is a review from 9th grade and HL1 SL material. The only thing that I want to emphasize, though, is that we are looking at Mendelian genetics with unlinked genes, genes that happen to be on different chromosomes. Okay, so that's it for dihybrid crosses.